On a winter night off the coast of Hokkaido, small fishing boats return to shore with holds full of silvery Alaska pollock. Yet what turns this story into a fascinating scientific snapshot is not the seascape, but a modern kitchen thousands of kilometers away where a child is nibbling on a crab stick and asking, "Mom, why does it look like crab, but isn't crab?" That seemingly simple question touches on one of the most sophisticated techniques ever developed in food technology, the reverse engineering of protein structure. The crab sticks we eat light, slightly sweet, and springy are not products of deception. They are the result of a carefully designed chain of biochemical interventions, executed with a precision comparable to reprogramming a miniature genetic code. Here, whitefish meat is not merely processed, it is reprogrammed. Protein chains are dismantled like a machine taken apart, then reassembled into a new architecture that mimics the elasticity of real crab meat, convincing enough to stimulate both human mechanoreceptors and taste receptors. This is not fake food. It is a biological material created not to replace crab, but to demonstrate that the material structure of food can be intentionally redesigned. To understand surimi, we must begin with myofibrillar proteins, the primary structural components in the muscles of any fish, in Alaska pollock. These proteins exist as actin and myosin filaments bound into actomyosin complexes, a system of molecular pulling machines responsible for muscle contraction and relaxation. The production of surimi begins by extracting these proteins from the original muscle tissue. When the fish is minced and repeatedly washed in cold water, a process known as leaching occurs. Fats, soluble enzymes, and odor-causing molecules are removed. What remains is a purified protein mass like the skeletal framework of the original mechanical structure. Notably, at this stage the proteins are still unformed. They resemble loose polymer strands awaiting activation. Low temperatures help prevent denaturation, a process in which hydrogen and ionic bonds break down, causing proteins to lose their natural tertiary structure. When this protein mass is pressed, kneaded, and mixed, the molecules begin to rearrange. Weak bonding regions separate, creating new anchoring points in preparation for gel formation. This is when surimi is born, a protein matrix capable of stretching, interweaving, and most importantly, being reshaped into virtually any desired form. If natural protein is like a brick wall, surimi is brick ground into powder and rebuilt into an entirely new structure with distinct mechanical properties. What makes surimi convincing is not just its ingredients, but its structural dynamics, the way molecules move, bond, and behave under the influence of heat, salt, and additives. During the gel formation stage, surimi is mixed with starch and sometimes egg white. These components act as cross-linking agents. In molecular biology, cross-linking refers to the process of connecting two polymer chains through intermediary bridges, forming a more stable network. When heated, starch swells and fills the gaps between protein strands, like reinforcing beams inserted into a structural frame. Egg white contains ovalbumin, a protein capable of coagulating into a fine network when heated, further supporting the gel structure of surimi. Yet the true subtlety lies in elasticity. Real crab meat has long muscle fibers that create a layered separation when pulled apart. Surimi cannot fully replicate natural muscle fibers, but it produces a new sensory equivalent bounce. Bounce emerges when the protein gel network has just enough cross-link density to be elastic, but not so dense that it becomes rubbery. This is an art of dynamic balance. If the bonds are too weak, the gel collapses. If too strong, the surimi becomes rigid and loses its ability to simulate real texture. By adjusting starch ratios, setting temperatures, and mixing speeds, food engineers can tune the chewing experience much like tuning sound, enhancing elasticity, reducing brittleness, or even simulating the fibrous pull of crab meat by pressing surimi into long, parallel strands. A crab stick does not only deceive the hand and the tongue, it also deceives the nose. To achieve this, the food industry relies on a complex system of aroma compounds. Shellfish extract contains molecules such as trimethylamine oxide, TMAO, nucleotides, and naturally occurring amino acids. When heated, TMAO breaks down into trimethylamine, a signature seafood odor. This is a chemical reaction based on the cleavage of the NO bond within the molecule, releasing volatile amines. To prevent the smell from becoming overpowering, Surimi uses carefully controlled amounts. Artificial flavorings are added to create a crab-like sensation rather than fully replicate the aroma of real crab. This explains why the crab stick flavor has evolved into a distinct taste category of its own, not identical to that of natural crab. Sorbitol, a polyol, functions as a humectant. With a molecular structure rich in hydroxyl groups, sorbitol binds tightly to water molecules through hydrogen bonding, helping the surimi gel retain moisture and protecting proteins from denaturation during freezing. Salt serves a different role: ionic stabilization. Sodium ions, Na-, reduce the repulsive forces between charged protein filaments, allowing them to draw closer together and form a more stable gel network. Salt acts as an ionic key that locks the protein matrix in place. Through these adjustments, each surimi stick becomes a chemical symphony, a hint of real seafood, a degree of simulation, and a great deal of precise calculation in how aroma and taste are activated. Yet flavor simulation alone is not enough. Surimi must also look like crab. This is where vision becomes a battleground, just as important as biochemistry. Crab sticks are coated with a red outer layer, typically using carmine, derived from cochineal insects or paprika oleoresin. These pigments adhere to the protein surface, producing the characteristic color reminiscent of snow crab legs. The process resembles food. Printing surimi is extruded into blocks, sliced thinly, then passed along a conveyor where color is applied. Some factories even create striped patterns to mimic the fibrous muscle layers of crab, a form of biological graphics applied to food. When consumers see the red exterior and white interior, the brain immediately activates familiar seafood recognition patterns. In other words, visual perception makes its judgment before the tongue even enters the experience. This form of simulation is far from cheap imitation. It represents the pinnacle of food biomimetics, the science of biological imitation in food design. The goal is not deception, but the recreation of experience using protein structures as programmable materials. Crab sticks are not a poor copy of real seafood. They symbolize a new way of thinking. Humans are no longer just cooking food. They are designing edible materials. Surimi demonstrates that proteins can be disassembled and reassembled like a machine, forming new structures with new functions. In an era when food systems face sustainability challenges, surimi stands as evidence that science can expand the value of the ocean by mastering molecular structure. So when you bite into a crab stick, you are not eating fake food. You are experiencing a work of molecular engineering where taste, mechanics, and chemistry converge within a perfectly reprogrammed structure.